Welcome to Guidance of Ultiplex-multiplex PCR Primer Design website. What is Ultiplex? Ultiplex is a primer design website. If you don't know what primer is, please leave. Ultiplex is a multiplex PCR primer design website, and it is automated. Ultiplex is a multiplex PCR primer design website. It could design primers for 1 to 100 targets, and all primers are reacted in one tube. Ultiplex is a PCR primer design website. It could design primers, filter primers, delete primers which contain harmful second structures, such as hairpin and dimer. Delete primers which contain SNP, or located in repeat sequence. Delete primers which have nonspecifical amplicons. Ultiplex also can cluster primer pairs, guarantee there are no dimers or nonspecifical amplicons between primer pairs of different targets in one group. Ultiplex also is an automated primer design software. For beginners, you only need one click to finish primer design. For experts, but you can define parameters by yourself. Ultiplex provide user-friendly web-based interface and informative results. You can adjust your input parameters to improve your design results. Generated primers has good quality. There are no unspecific amplicons in the experiments of 108 single output primer pairs. Each pair only have one clear amplicon. After 2 candelas round PCR, products are at the adequate range for NGS and could be sequenced directly. To saving times, you can pause here and choose times you're interested in of this catalog. However, if you don't understand functions in some chapters, you may need to check those skip chapters. Basic functions are listed on the earlier chapters. Background knowledge. How to design primers for one target. Ideal primers should be sensitive and specific in PCR reaction. Sensitive means primers could amplify products in high efficiency. Factors affected primer efficiency, such as hairpin, dimer and SNP in the ends of primers, should be eliminated. Specific means primers could generated non-specifical amplicons. Those primers and those primers located in repeat sequence should be eliminated. How to design primers for 2 targets. In addition of demands for each target, dimers between primer pairs of 2 targets should be eliminated, and also those nonspecifical amplicons between 4 primers on genome should be eliminated. How to design primers for more targets. In addition of demands for each target, dimers and nonspecifical amplicons between all primers should be prevented. If there are 20 targets in one reaction, 760 times validations will be needed to check the dimers and nonspecifical amplicons. When the number of target raised to 100, 19,800 times validations will be needed. Experiments could not finish these validations in short times. Ultiplex provide automated multiplex PCR primer design functions and could finish large number target primer design. Introduction of layouts. New project layouts are showed as followed. At first, input your project name. Please make sure your target name not in our database. Then you can input your target's informations. This software design primers based on the genome coordinates of your targets. Targets informations should be in 4 columns, include chromosome name, start coordinate, end coordinate, and your target name. Columns can be separated by space or table. You can paste your bed formatted strings in the input box or upload by files upload box. After filled informations, please make sure upload your target information. Please make sure upload your target information. Website will check the format of your input information. After your upload, website will show total number of your uploaded targets. You can input parameters for primer design, include product size, primer TM value, GC content, primer numbers for each target. Bigger numbers will cause slower design speed. Target reference, coexist reference. Input primer filter parameters. To filter primers contained hairpin. To filter primers contained dimers. To filter primers contained non-specific amplicons. To filter primers contained SNP. To filtered primers contained repeat. You can choose whether add 2 candelas round PCR tags. After that, press Start Design. After your submission, it will be redirect to Results interface. There is a results link on the top of the page. This is the only way to check your primer design results in 7 days. Please write down this link on your notebook or your computer. Followed with update and progress information, target information, parameters for primer design, filter parameters. Summary of primer design results will show the success rate of primer design, as well as success targets number and fail targets number. In the drop-down menu, you can select one failed target to check the detail failure reason, include GC content failed, too low or high TM. Long poly XSEQ, unacceptable product size. For example, in this project and for this target, there are 225 forward primers, 301 reverse primers, paired primer pairs 67,725, but all primer pairs have unacceptable product size. As we dig deeper, we could found this target cover 400 BP in the uploaded bed information. However, primer design parameters require products smaller than 300 BP. You can adjust your input information to improve your design results. The process bar show the real-time progress of your design project. Finished results in each phase will be showed on this page, and unfinished results will be showed after finished. Summary of primer filtration will show numbers of primers which have hairpin or dimers, numbers of primers which located in repeat sequence or SNP, numbers of primers which have non-specific amplicons, and numbers of final qualified primers. You can check all targets through scroll bar. You can adjust bar area to zoom pictures. For example, this target has 84 primer pairs located in repeat sequence, 7 primer pairs contained hairpins, and 8 primer pairs are qualified. If one target has no qualified primers, the results will emphatically appear in the summary of primers of failed targets. According this summary, you can adjust your input information to improve your results. In the detail results of qualified primers, you can choose single target to download selected target information. You can also download all targets primers. You can also check details of qualified primers of selected target include primer penalty, primer sequence, primer location and so on. You can also check chromosome locations of primers of selected target. The unity and incompatibility between different pairs are tested for each pair, and compatible pairs are clustered. The list of maximum unifiable and compatible primers is generated. The overview of genome location of compatible primer set will showed as a Circos map. You can also check details of primers of compatible primer set include primer penalty, primer sequence, primer location and so on. Download whole multiplex primer set. You can synthesize primers with downloaded files. Example: primer design with human HG38 reference 1 click. Input your project name must be unique first. Then paste your target information. Upload. Keep other parameters as default. And you can start your primer design. Primer design with human HG19 reference. Input your project name must be unique first. Then paste your target information. Upload. Select HG19 in target. Select SNP_HG19 in skip sites drop-down menu. Select repeat HG19 in skip area drop-down menu. Add tag of 5 ends. Keep other parameters as default. You can start your primer design. Skip self-defined sites and HG38 SNPs. There are sites, SNPs and other small variances researchers need to skip for primer design. In this system, human SNPs are stored previously. If you want to skip sites for your own purpose, you can use this function. You can choose whether involved system human SNPs Input your project name must be unique first. Then paste your target information, upload. Select SNP_HG38 in Skip Sites drop-down menu. Click on the Choose File button, choose your own 3-column bed file. Click on Open button, click on Attach button. Now you finished your own site's file uploading. In the drop-down menu, the name is your own site's file name and HG38 SNP name. Keep other parameters as default. You can start your primer design. Only skip self-defined sites. There are sites, SNPs and other small variances researchers need to skip for primer design. In this system, human SNPs are stored previously. If you want to skip sites for your own purpose, you can use this function. You can choose whether involved system human SNPs. Select Null in Skip Sites drop-down menu. Click on the Choose File button, choose your own 3-column bed file. Click on Open button, click on Attach button. Now you finished your own site's file uploading. In the drop-down menu, the name is your own site's file name. Keep other parameters as default. You can start your primer design. Skip self-defined areas and HG38 repeat sequences. There are areas, repeats, tandem repeats, indels researchers need to skip for primer design. In this system, human repeat sequence is stored previously. If you want to skip areas for your own purpose, you can use this function. You can choose whether involved system human repeat sequence. Select Repeat HG38 in Skip Areas drop-down menu. Click on the Choose File button, choose your own 3-column bed file. Click on Open button, click on Attach button. Now you finished your own area's file uploading. In the drop-down menu, the name is your own area's file name and HG38 repeat name. Keep other parameters as default. You can start your primer design. Only skip self-defined areas. There are areas, repeats, tandem repeats, indels researchers need to skip for primer design. In this system, human repeat sequence is stored previously. If you want to skip areas for your own purpose, you can use this function. You can choose whether involved system human repeat sequence. Select Null in Skip Areas drop-down menu. Click on the Choose File button, choose your own 3-column bed file. Click on Open button, click on Attach button. Now you finished your own area's file uploading. In the drop-down menu, the name is your own area's file name. Keep other parameters as default. You can start your primer design. Self-defined genome reference, not human. This website only uploaded human references previously. If you want design primers for plants, other animals and microbe, your reference sequence need to upload first. Click on the Choose File button of Target Reference. Choose your own faster format file. Click on Open button. Click on Upload button. Reference files must be faster format. If your file is bigger than 10 MB, please contact with admin. Please make sure the chromosome names in Target's input box are in uploaded reference files. Now you finished your reference file uploading. Keep other parameters as default. You can start your primer design. Self-defined coexist reference, human gut microbiota primer design. Researchers may need to design primers for several species in complex samples. There are similar sequences in different species' genome. Unspecifical amplicons and dimer between primers of different species need to be eliminated. You can use this function to define coexist reference. Select Desire Reference in Coexist Reference drop-down menu or upload your self-defined coexist reference file. Input other parameters according to your needs. And then you can start your primer design. Thanks for your watching.